Hey guys, so today we are going to try to understand prokaryotic cell. So let's get right onto it. So before we start to talk about prokaryotic cells, we know probably that they comprise bacteria and archaea and they come in a lot of different shapes. Sometimes they just look a lot like spheres, so they are spherical, and when they are that, we call them coccus or plural cocci. Sometimes they are rod shaped, we call them bacillus. They take very funny names. Sometimes they look like comma, right? And then you start calling them vibrio. Sometimes they look like a spiral, and that makes sense. Let's call them spirula, right? Now, sometimes they do not have any shape at all. They are very pleomorphic. They can change shapes, and the example of them are spirochetes, right? So as you can see, there's a lot of weirdness going on in bacteria. Now, actually, till the point of time, you must have read a lot of things about bacteria or prokaryotes in general, and you'll see that they have a lot of exceptions. So we'll start with the basic structure. So prokaryotes generally obviously have a plasma membrane, right? Now, the better term I would use right now is a cell envelope. So because they might have a lot of stuff, so cell envelope is a term that is used for all layers that they might have, starting with plasma membrane. So that is why plasma membrane plus. So plasma membrane and all other layers that they might have. So besides plasma membrane, they have a cell wall. Now, this cell wall is not like those of plant cells. It's made up of a complex polysaccharide we call peptidoglycan. Peptidoglycan have sugar derivatives. Each is made of unit called NAG and NAM, and there are peptido, that means peptides linking several things in between. Now, based on the arrangement of cell wall and membranes, the bacteria can be gram positive and gram negative. We'll talk about that in another video. But besides having cell wall, they might have some other layers as well. Now, if that other layer is very organized, you call them capsule, which can be carbohydrates or and proteins. If it's not very organized, we like to call them slime layers. Now, there is some sort of extra name that you might have heard, which is very much related to that, and that is glycocalyx. Now, again, glyco, carbohydrate layers that might be used for both slime layer or capsules. Now, why they have these many layers? Now, as you can understand, they have to do a lot of osmoregulation, right? So cell walls, they provide structural strength, right? So they provide a lot of structural strength for osmotic variations that might come here, whereas these slime layers or capsules, they help them avoid being eaten, right? So they are there to give them some kind of protection against phagocytosis, right? So they give you some sort of protection from phagocytosis. Now, so these are a few layers that you should be aware of. Now let's go to some of the stuff that's more there on the uh, outermost layer, cell envelope. So remember, these are all parts of cell envelope. So plasma membrane plus layers there, that's commonly called as cell envelope. Now, to move, the prokaryotes many times have structures called flagella. Now, flagella are structures of motility, that is movement, and they generally have a part that's called as a motor injected into the plasma membrane in the cell wall. Then they have something that's called hook, and then finally they have something that moves called filament. Now there's a very key difference between eukaryotic flagella versus prokaryotic flagella. A prokaryotic flagella is without the covering of plasma membrane. Eukaryotic flagella, like in the case of human sperm, they're always covered by plasma membrane. Okay. Now, the flagella can be placed in a lot of different ways. They can be placed just around one end, right? Or they can be placed all over, or they can be placed at opposite ends. All such variants do exist, but we won't be going into the detail of the various names the bacteria takes based on that arrangement. Now, then there's a very funny structure here that is again sometimes used very interchangeably, and that is fimbriae and pili. Now, remember that the fimbriae is generally used or is always used for attachment. This is how the bacteria hooks onto your surfaces and holds onto the inner layers. Pili is generally used as a transfer tool. Now, much unlike humans, the bacteria or simply prokaryotes, they can transfer even their genetic material. So this is why they are very successful. So if one bacteria comes up with a gene for, let's say, antibiotic resistance, it can transfer it, right? And that brings me to its important part, and that is the DNA. Now, bacteria, their genetic material is most cases a circular chromosome. Now, when I say circular chromosome, remember they do not carry histones. They are non-histone proteins that allow them to condense their DNA. Now, you have to understand that generally the genetic material, the length of the genetic material is around, let's say, take example of E. coli, it's around sometimes 300, 400, even 700 times the length of the bacteria, right? So you need some sort of proteins to help them fold, and sometimes they aren't proteins, they are just folded around the DNA itself, right? Now, can they have more than one chromosomes? Well, generally not, but yes, exceptions do exist where you find more than one chromosome. Okay, but that's not the only piece of DNA that they have. Very funny, they have some small, very small, non essential pieces of DNA, again circular, that's called plasmids. And this is where the bacteria gets really naughty. So, the genes for antibiotic resistance, or let's say temperature or heat resistance, that kind of gene is stuffed in here. They might also have the gene for formation of pili. On the basis of that, the bacteria can be an F plus, they have the pili forming gene, or an F plasmid, or they might be called an F minus. So you see that they have a lot of mechanisms. They believe in sharing is caring, right? Now, what else is important to know about bacteria? Functions. So bacteria, where do they perform all the functions? So just outside this zone, remember I am just calling this a zone. This zone where they have the genetic material is called nucleoid. There is no membrane, it's just a zone. But again, as I said, it's bacteria. You want to find weird, they'll give you weird. So in some exceptionally different bacteria, they might even have some sort of membrane or protein covering around the DNA, but generally they do not have, they just have a zone. So whatever is left is the cytoplasm. Now, cytoplasm is the seat of all chemical reactions that happen. That means that they do not have membranous organelles. So no membranous organelles are found in uh, bacteria, right? But again, as I said, exceptions. Some compartments, no membranous organelle doesn't mean they do not have compartments at all. Some compartments do exist. Generally, they are made up of proteins. Sometimes they might be even of other substances, but generally they are made up of proteins, and they may carry some different stuff, different types of reactions. So sometimes they are equipped with detecting the magnetic field, and sometimes they metabolize ammonia. So all those exceptional structures do exist. Now, obviously they need more compartmentalization because they're very efficient, right? So as you can see in here, the plasma membrane folds inwards to form these pockets. Sometimes you might get a name for these pockets called mesosomes. So these pockets essentially localize a lot of enzymes which help in uh, carrying out several reactions. Now, when the prokaryote does not have a very good atmosphere, it goes and forms a very special kind of structure called endospores. Now, endospores are extremely resistant structures that help the bacteria survive the odds, and uh, they again reproduce to give the bacteria when the uh, nutritious surrounding comes in. So this was what we learned about bacteria. We started with the plasma membrane. We then talked about the various other kinds of layers. We talked about the cell wall, G plus, G minus, gram positive or negative, the staining method given by Christian Gram, which we'll discuss in some other video. We'll talked about the capsule or the slime layer, which gives some sort of protection. We talked about appendages, that is our own flagella, and we also talked about fimbriae and pili, each having their own role. We talked about the genetic material, which is generally the circular chromosome, but it can be linear as well. Remember, bacteria, you'll always find exceptions. It can be linear as well, some cases. Then you can even have more than one chromosomes. You have nucleoid, you have cytoplasm. uh, there are no membranous organelles, but sometimes you do have compartments made of proteins for different work. Sometimes you have mesosomes, the pockets formed, which increases the sort of localization that you require for processing. So I hope this whole diagram makes sense.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.